So, a quick introduction to Kirti. So, I'm sure many of you know him. He seems to, we were just sharing that he's been haunting my life for the last 6 years at least. He completed his PhD in 2016 and has gone on to do many different things, including the what he's going to talk about today, which is single molecule imaging using mercury lamp as a light source. So he's did his PhD in Heidelberg in the lab of Christoph Kramer, and seems to have worked at least with a number of Nobel Prize winning microscopists, and if nothing else, his contacts seem to be quite extensive in that range. So, and I think that's probably all I want to say about Kirti. So I was going to keep it short and snappy, and over to Kirti.
Thanks, Alex, for a nice introduction. Just to say that I haven't worked with a Nobel Prize winner. So Christoph Kramer, Joe Gall, none of them actually won Nobel Prize. So, but I know a few of them, and I'm afraid that there are some demons coming. So you might be haunted during the talk. Just want to make sure if I'm audible and everything is fine, and then yeah, I just continue. So the talk is divided into two parts: MINFLUX and laser-free super resolution. As you know, this Imaging ONEWORLD is very method focused. So instead of starting with MINFLUX, I will start with SuperRes and then go on. So as some of you know, like I put out this cheesy question on Twitter, like which one is MINFLUX? You have two sets of dots, right? And I put out a quiz. The funny part was, I will come back to all the details, what I did, what processing I did later on. But the funny part was, roughly 56% of the people got the wrong answer, right? So in just one tweet above, I had this image. And if anyone would have seen, this is from Jonas Rees' lab. And there is the citation right there. And this is just rendering this image with different precision. And then I had a one MINFLUX image, which I directly took from the Hell Lab paper, the Nature Methods. So yeah, surprisingly, either people don't read the tweets or whatever. I think, yeah. So this was one trick. So left was the correct answer, and this image, if you see, is the same. But we will come back to this image again and talk about a lot of filtering, processing, how to image dense structures. So there are three aspects to this talk. First is elements of single molecules. Some of the people in the audience, I know, they are experts in the field, but I still thought, since this is a method-focused talk, I will just give you some elements. And these are also important as we go along to understand the minutes of single molecule and also MINFLUX. Then I will jump into 10-15 minutes on a method I developed. And then finally, MINFLUX. And I'm also happy if your questions are relevant to the talk, I'm happy to take them during the talk also. So no problems. So this is, so single molecule methods. There are basically three ways you can do it: either having a chemical buffer, or switching the fluorophore, cis and trans with light, or you can do binding and binding kinetics. So usually in classic Alexa 647, you'd have zero background. And what I'm showing you is a cell nucleus labeled with hooks. And what you see is binding and binding kinetics. So it's a bit like DNA paint, except that over here you use the DNA itself to do the binding and binding kinetics. So you see all these dots coming up, and you have background in this case. In classic Alexa 647, what people use, you have almost zero background, but then you can subtract the background, find the points, and if you image long, you will sum this and you will get a high resolution structure. So this would come into this category of when DAPI binds to DNA or Hooks binds to DNA, there's 1,000-fold enhancement in fluorescence, and this is the example of that. So we use this to study a lot of different things. One of the examples was the discovery of a new reversible chromatin domain. So this is a cell nucleus. You see this classic heterochromatin at the periphery, then you see these chromocenters. So when we stress the cells, we made a special chamber where we devoid the cells of oxygen and nutrients, and it formed these special ring-like structures. And when we put the cells back into normal conditions, it reversed back. So this was one of the major findings that we had with this Hooks-DAPI method. I won't go into the sample prep. This talk is mostly on microscopy. And if I have to tell you like two most important things in single molecule, is the interplay between precision and signal density. I would emphasize that I think the most important aspect is sample prep, but that's another thing, and maybe in another talk I will cover about it. So what do I mean by signal density, right? So you have the target protein and you have the fluorophore which you detect, and the fluorophore you detect is the signal. So and this signal is because the fluorophore is bleached. So it's really important that you image very long, right? So let's say like as you remember, you saw these points coming on and off, right? So if you only have 1,000 points, you won't be able to make out the underlying structure. But 10,000, you start to get something, 20,000, a bit more, 25, 50. And if you see, like, if you have 100,000 signals, you will be able to make out a structure. And this is really important because most of the things that we do are between, let's say, 20 to 200 nanometers. And you don't know that underlying structure in advance because this is all new biology, right? So having very high signal density is super important. And you have to complement it with good precision. So for example, if I take the same number of points, let's say, 25,000 points, and if I have 2 nanometer precision, image looks something like this. But as you increase, like with 10 nanometer, you see more features, right? But if I keep increasing the uncertainty and precision, I'm using both interchangeably. So you see, like with 100 nanometers, it's more blurry. So why is this 10 nanometer looks better than 2 nanometers? Because in this case, we are blurring each molecule with a broader kernel, let's say a Gaussian, and then it's covering up for the missing points. So you see like how signal density and localization precision goes hand in hand. So if you really have a lot of signals, you have to also keep on improving with precision. So this is one of the important aspects in imaging, and this is what we behind the scenes try to optimize with not super high laser power, because if we have too high laser power, we might get very precise points, but we might bleach the sample, right? So we try to have the best laser powers, best integration time for each molecule so that we can somehow come in this domain. And most of this thing comes with experience. Like the more you image, the more you get the feel for the sample, how long the bleaching will last. Another thing is the localization precision is not image resolution. So I told you there is a protein, right? And then there is either a primary, secondary antibody, and then a fluorophore, right? So there's a difference. So you can have a resolution of the fluorophore or resolution of the target. So what the target is what we call localization accuracy. So let's say what you see in a star is my protein of interest, right? And then we have a primary, secondary antibody, and this is the fluorophore. So we are detecting something this on the camera, right? So this is a case of very good localization precision. Points are clustered nicely, but they are away from the target. In this case, you see like points are closer to the target all over, but they are spread. So it's a very poor precision, right? And this is the ideal case where you have a precision and also points clustered to the target, and then you can filter out. I will talk about filtering in a minute. So now coming to one of the methods I developed, I call it laser-free super resolution for time being, because you will know in a bit. So classically, there are few things, microscope setups are big. What you see over here, they are very complex, most of the single molecule setups. And I think there is one in Oxford in Micron, which I saw, it almost takes 2 rooms. So this is OMX. So most of the microscope setups are big. And usually the underlying thing is most confocal wide field setups look like this. And then as you go for light sheet or more advanced like SIM or single molecule, you find more optical tables with microscopes spread up. So this is a small project which I did in US. So when I moved to US, we had a STED microscope for which we needed a 6 months confocal training followed by 6 months of STED training. And then I had this a very old Olympus BX61, and I thought, like, can I do super resolution on it? And then, like, what you really need to do single molecule, right? You need a very good camera. Then you need a laser. Classically, this is what people did, because back in 2000s when we had GFP, it emitted around 200 photons, right? And then you use laser so that you can extract maximum photons. Over the years, all these Alexa dyes have got super bright, and you don't need the lasers anymore. So actually, it was more like this setup I had, and what can I make of it? It had lamp, and there was no funds to buy lasers. So I thought, can I do something with the lamp, right? So one of the interesting things with lamp is it has these several peaks, right? So around 560 and 400 nanometers. So the idea was, can I use this 560 peaks to, let's say, excite 594 die, and then can I use this 400 to reactivate the fluorophores, right? So this was the idea I started with. And rest of the things, I managed to get a high NA objective lens from the core facility and things like those. So this is how the setup looked like. So this is, as I said, this is BX61 Olympus. Joe Gall bought it in 1998 for some $10,000-$20,000, and no one was actually using it. It's a very old one. The computer, you see, it has 2 GB RAM, and I attached a rod and some this to just, you know, think for stabilization purpose. The problem was, if anyone would even come in the room, just remove the curtains, the sample would move. So eventually the rod and bit over here didn't work. The only thing worked was going 12 in the midnight to do the measurement when there was no one in the building, and then to account for the stability of the instrument that you need for single molecules. So a lot of small optimizations, I dark lamp, fitted a new one, so small things like those. But the idea is you can really do single molecule stuff with a traditional microscope that's present, and even you don't need very sophisticated computers. So this is just an image showing how single molecules look like. So this is how you see, right? You have a field of view and you have single molecules coming on and off. And this is the spectra. You see they are bleaching a bit slowly. And then this is what final result. So basically these are mouse meiotic chromosomes. So these are 2 homologous chromosomes, the paternal and the maternal. They are synapses, and you can't resolve them with confocal, but then you can resolve them nicely with this own setup. One of the things was, I told you that we could use this to reactivate, right? Even with lamp, there is some bleaching, right? So what I did is I changed filters, right? From Trixie to Darky filters. So I used these wavelengths kind of to reactivate. And one of the very interesting thing, I personally feel this is key to a lot of photophysics that I hope I am able to explore in future, is that I could keep changing these filters and image, like, I think I imaged close to 10 to 12 hours nonstop, you know, changing from right in the midnight to something 10, 11 a.m. in the morning on a weekend. So this is another thing, like, you know, what is photo bleaching? I think we need to revisit some of the classic concepts of photophysics. Another thing, what I did is the people at Carnegie were skeptical that, you know, even though these mouse meiotic chromosomes have very well-defined structure, and they were skeptic if this structure is really true, right? So in the process, what I ended up doing was a sequential correlative super resolution. So what you see is, again, confocal. Then I did a STED. So we had a Leica SP8. So these are the prices that below. So confocal typically is these days like $400 to $500. STED is around $900, and this is around 10 to 20, right? So there are several things in this, right? So one thing is, I think this has to be kind of the future of microscopy, doing some sequential correlative imaging. Because what you identify with one method, you have to reestablish the structure with another method, right? So what do you see? First, we see there's a lot of consistency. There are 2 strands. So at that level, we are sure, okay, there are 2 strands in this protein, we are damn sure. Another thing is we see, oh, these strands are tied at one end, right? So why so, right? And there's more concentration right over here and there, and then over here. So at some level, there is structure similarity. But then you see some of these dots in the background, which, okay, these are some molecules. Then you see something dense over here. So you see, like, by doing such sequential correlative imaging, I can safely claim that these proteins have 2 strands, and each of them is roughly 60 nanometers. The distance between them is, for example, 100 nanometers. So these things I can claim. But then can I claim if this dot is real? No. So this is why some sequential correlative imaging is super important. One of the things that really came out of this, I started to wonder why one end of this protein are far apart while others are joined. So I looked into literature and I found there are different histone modifications. So apart from microscopy, my other passion is in chromatin and chromosome structure. So histone modifications are my favorite. So I kind of know that mouse chromosomes, they are actocentric. So for example, the telomeres, the centromeres localize at one of the telomeres, not at both. So that's why these ends are joined. And there is this classic K9me3. It's a mark of centromeres, and it co-localizes at one end. This is an active histone mark. So it's more away so that it's accessible to Pol2. And then we have a K27me3, which forms these very symmetric clusters along the chromosomes. And this is again the Hooks-Darky stuff, right? So this was very cool. So if you Google on, Google is called a structural meiosis, you won't find many articles. And I believe like by doing this correlative imaging, we can not only discover structures, new things, but we can also cross-validate, right? There's a lot of DNA at one end, and now we understand why it co-localizes at this end, right? So there's a lot of behind-the-scenes cool biology that we can do. And another point I want to make is a lot of, let's say I mentioned STED is, I don't know what's the current price. Few years back, it was $900,000. But you can make very cool setups, right? With very little. So Johannes Hobline, like he maintains all this list of open source software, and most of these setups you can build for 50 to 100,000. So yeah, even I showed with 10, 20,000, you can do a lot of cool single molecule imaging, research grade. And another thing I want to emphasize again here is, like the research fund is zero sum game. So right, like if ERC or NIH gives away $2 million for something, they said, oh, we just gave. So if one method takes most of the things, you get limited resources, right? So now I come to the next talk, which is on MINFLUX. And I guess this is why a lot of people are there in the audience. I actually don't know how many are there, whatever. So let's talk about MINFLUX. This is the exciting bit. I kind of tempted many STED and MINFLUX people to come to this talk. I hope they are there. I can have a quick look who is here, but yeah. But I see like there are at least like 6 pages of people on Zoom, so which is very nice. So let's start me with a disclaimer, right? So there is no raw data in the paper because yeah, it's a proprietary software. Understandable. I also wrote to Stefan, no response to my emails. I asked him to clarify some questions and data, none of it. So Nature is pushing for a lot of, validation of methods and easy accessibility to data. So I tagged Rita Stark, and I think she has been very kind and she has been helping to get access to the data. And let's hope I have the raw data at some point. So what I will be showing is the data or the images that are in the paper and what we can learn from it, right? So first of all, there is not only MINFLUX, there are at least like 5 to 6 methods doing, let's say, direct combination of STED or single molecule or SIM or single molecule. Basically, getting single molecules with patterned light. It can be a donut or what you have in SIM. So there are a number of these methods. I think there has been effort by Göttingen people to club it under MINFLUX, but if Carlos and Sandrine and others agree, we can all call it MINFLUX, no problems with it. So what is MINFLUX? Right, so I talked about single molecule and STED. So this slide is actually a snapshot from Stefan's Hell Talk. If you type this on YouTube, this is the link. And basically what he talks about is combining the advantages of STED, which is, a lot of photons from the laser. Basically by STED here, he means the donut. And then combining the single molecules, switching coordinates that you get from Palmstorm. So if we look more in detail, I won't talk about this because this is not my area. I haven't developed MINFLUX, but I'm just briefly for a minute or two touching what the concept behind MINFLUX is. But more I will focus on application side rather than technical side. So MINFLUX is basically like, it uses the STED beam, or let's say the donut beam, not the STED beam. And you have like almost zero laser intensity at the center, and then you have a peak intensity, right? So it's basically, if you have a fluorophore right here, right, you will have no photons. So it's basically localizing fluorophores by not seeing photons. So it's a very cool idea if you think about it. And there is a Gedanken experiment, which I will show in the next slide. So basically what you do is you scan the position of a fluorophore and you end up with this quadratic approximation. And basically you can fit it and then find where exactly a fluorophore is. So for me, it seems like a computational maximum likelihood problem, guessing the position. So it's a combination of, I would say, the donut beam with single molecules with some likelihood estimations. And what Stefan emphasizes, it's wavelength independent, and also it just depends on the L, right? So you almost need 0 photons in an ideal experiment. And this is what he writes in the paper, actually. So one of the things I really like about Stefan is how much, Stefan Hell, I mean, is how much effort he puts in writing papers. And these are like great readings. So when I was first reading this paper, and I thought, wow, like I haven't read a text like this in the paper. So something like this, unfortunately, it is impossible for us to place a donut 0 right at the molecular coordinate in a single shot, which is why perfect localization without emissions can only be performed by a supernatural being, a demon, who knows the position of the molecule in advance. And, I have some philosophical inclinations myself. So I know, like, in classical, let's say, European history, a lot of scientists, Descartes, Laplace, Maxwell, Darwin, they use demon for these, like, this in the thought experiment. So this is what he goes on. Yet this Gedanken experiment suggests that approaching molecular position by targeting the 0 of excitation donut to the molecule should reduce the number of detected photons required for localization. So yeah, it's a nice thought experiment, and there has been history, and I believe 100 to 100 years from now, Stefan Hell will be recalled in the same league as these great scientists. And of course, if we don't play gods and demons, then who will, right? So let's now focus on the application. So that was all the theory I wanted to say. And now let's see what we get out of MINFLUX. And this is, if you haven't heard me until now, if you focus from now, this is the most important bit, right? If yeah, for whatever reason you were not focused. So we will be talking about nuclear pores. And this is how nuclear pores look like. So this is an image from Jonas Rees, right? So this is a EM density map. So nuclear pores have basically 8 subunits, and each of these subunits have 4 copies. So 12 nanometer is the roughly diameter of each subunits, and there's a spacing of roughly 40 nanometers. And then there are 2 layers of these pores, which are 50 nanometers apart. So this is the let's say the playground we will be playing in a bit. And this is the main image from this is figure 2 from the Nature Methods paper from Hell Lab. And what you see is these nice images of nuclear pore, and then you take this image and you see. So over here, I will show you the zoom, and you will nicely see roughly 8 components with these individual copies of these 4 subunits. So let's look deep, right? So I have this image. So if I take this is what it is. We roughly can count maybe 1, 2, 3, 4, 5, 6, maybe 7 if you want. And maybe this has 4 copies. This might have 3 copies. And then I zoom in this one, right? And then maybe, So the point I'm trying to make is, how do we zoom in from here to here, right? So what I did is I took this image, I put it in Fiji, and I downsampled it. I basically had to downsample it 63x times to really get back to this same level of, resolving the molecules. So you see, you can resolve these dots. This is the whole big field of view. And then I had to really, so do you think this thing is zoomed in 63x times to this thing? Maybe we don't know, right? So this is what I'm inferring. There's no raw data. So this is the first bit. The second thing is, I think the most catching figure of whole MINFLUX is these molecular components, right? Now you have these subunits which you can't resolve, and you now see the individual molecular components, right? So this is the main highlight of MINFLUX, right? So I took this, right? So I will explain you how you get. So what you see over here is a lot of dots with same precision, right? So I will tell you how you get this image. So this is from the Nature Methods Hell paper, and this is from the Rees lab, right? And again, you see all these 8 components with same. So I don't know which exactly pore it is. So I was able to access the raw data from EBI website. And we will come, like, if molecular resolution is all about dots, and I will come back to this. Let me talk about another very important aspect in the MINFLUX paper, which is localization filtering. So as I said, there is no raw data. So this again is data from Jonas Rees lab. So if I take one of the cell nucleus, and this is roughly 500,000 localization, this is how the plot looks like of the precision. If I just take this one, you see a very nice bimodal precision plot. and then I zoom, and then I get like something like 5,000 localization, 300 localizations. And now if I only take localizations, which are less than 5 nanometers, right, I get this nice, very molecular image. And it's very easy to go from here to a very crisp looking dots, right? So before I come back to MINFLUX, let me talk about importance of filtering. So nuclear pores, as I said, they have, classically people think they have 8 components, right? So one of the problems in modern sciences, we are very biased that, you know, nuclear pores will have 8 components. There are studies from EM which shows they are like, you know, 7 subunit, 9 subunit pores. So so to put it softly, you know, sometimes in modern science, we are so much motivated by a structure. Let's say in this case, first of all, there is a circular fitting done to remove all the backgrounds. Then we have this rotational alignment where we are looking for roughly 4 copies of each subunit in each of these. And then based on that, we calculate effective labeling efficiency, how good our efficiency is. So my point here is, it might be the efficiency is good, you know, we had 7 components, but so our calibration is standard is 8 components, each with 4 copies. So this in turn kind of biases. So though the images, final images look very crisp and sharp with no background, like original images look very noisy and things like those, but in the papers it look, but then again, like we are very biased. So I think it's super important to always have the raw data going with the main paper. So coming back to the MINFLUX, as I said, we had this ring, right? So what I did now is I have these 25 localizations and I just blurred them with a 1 nanometer version. And now you see this super resolved points. And this is for comparison the data from MINFLUX paper, right? So yeah. So you see like, so the question here is, do you think all the points that we were doing our scanning with the donut have the same precision? So I think this is a rendering then. And the problem is not with the rendering. The problem is, as of now, I haven't been able to find a documentation either in the supplementary material or in the main paper where they said that they rendered the points with the same precision. So for me, this is really pushing, showing these dots to claim the molecular resolution, you know, that we can see the molecules. And I think this is borderline, you know, not good in the sense that, you know, either you document it that we intentionally, this is what single molecule people do for over the years. For visualization purpose, you pick up most 60-70% papers, you will find that for visualization, they blurred with the same precision. It's fine also as long as you do it, you know, you can almost filter. And another thing is the filtering, this is the supplementary material for, what the authors show is, you know, so they filter based on relative photon count. And then the second filtering is based on the background distribution. So they basically remove the points, right? And this is another super important aspect, right? So there are 2 levels, at least 2 levels of filtering in the paper that we know of. And you see like how we can go from a blurry looking subunits to a more refined subunits. And if I change my precision, I can even see the molecular components, right? So ideally, what should you do, right? Ideally, show me all the data, show me an image with all these points, and then you show what is the effect of removing this. Again, what is the effect of removing this, you know? So then I know, okay, these are really background and these are not affecting the structure itself, right? So this is another super important aspect. And we will come to this. Another super important is in nuclear pores, we know there are 8 subunits and each have 4 copies, right? So it's very easy to filter out and really go for the kill of watching these subunits with 4 copies, right? What if I don't know the structure, right? So, and last but not the least, in the MINFLUX paper, they don't provide any single molecule, any independent method that I was talking about, you know, sequential super resolution. One method showing the same structure, other methods showing the same structure. So without this, you know, like it's too much pushing the limits, right? And second thing is, if you just filter out and you take this population, you also improve the resolution, right? Because these are very well, you know, localized points. So in a turn, you also remove the precision. So let's say you don't believe whatever I say up until now, right? Because these are inferences drawing and I'm using data from another paper. By the way, Jonas Rees uses the same snap tag that Hell Lab used. So it's the same labeling, just the different detection. But let's say you are still skeptical. It's a big lab with 100 people, and my inferences are based on what I'm showing you. So let's forget that, and let's see if we really need molecular resolution, right? So this is EM. So nuclear pores were first described by Joe Gall in 1967. You can nicely see the 8 subunits. Marcus Howard did D-Strom in 2011. You can again very nicely see the subunits. Jonas Rees, I think, has the best localization setup in his lab. You can also very consistently see 7, 8, component or subunit pores. But when you go to MINFLUX, you can do the counting yourself, right? I didn't put numbers here to bias your thinking, but you can go home and count how many times. Like this is, I think, the best structure which has 7 components. The rest, if you look into the images, you don't even see close. Mostly are 5, 6 components. So first of all, you're not only missing the components. So let's not talk about number of copies in each component. Over here, you are detecting 5. Here, you are detecting 2. So what's the use of MINFLUX, right? If I can't figure out the underlying, how the 4 copies, are they organized in tetrahedron or they are just randomly distributed, right? So there is no comment how these copies are organized. So what's the real use, right? I can watch beautiful images just by changing the precision, right? So I don't need to have this beautiful images, right? So this is the first point. The second point, even if you didn't believe anything, right? So what happens when we do 2-color MINFLUX, right? So this is again D-Strom images from Marcus Howard lab. You see the outer ring, the inner ring is made up of WGA, and very good image, a bit noisy. Jonas Rees, as I say, he has one of the best images. You see the outer ring, 8 components, the inner ring of the pores, very nice. You go to MINFLUX, 2 colors, and wow, like 8 subunits, no components, the inner blob you hardly make out, right? So this is the second, like with 2 colors, you can't even make out the subunits, right? Let alone the molecular components that were claimed by the MINFLUX. So what's the big fuss with the method, right? Why shall I invest $2 million in this method if I won't be looking at nuclear pores, right? Another thing is, let's touch on the importance of structural information. So the only structure MINFLUX has convincingly show is DNA origami and nuclear pores, about which we have a lot of a priori structural information. What happens when we image structures, let's say postsynaptic proteins, which have no underlying structure, right? So this can mean anything, right? You can show me anything. You can show me 2.5 nanometers. What does it mean, right? Okay. Another important thing, no EM images, no other SIM or STED images that you complement. You show the same structure with the STED. Okay, I believe, right? STED MINFLUX, right? So this is another point. Let's look at another structure which just got in a PNS contributed paper from Stefan, right? So they claim they see this, the MIG60 forms a ring-like arrangement, right? You see a ring, okay, hollow tubular ring-like structure, but again, no other independent. So this is like nature methods, right? People buy it. I just heard one of my colleagues that MINFLUX is on his shopping list. There are 2 bought in Germany, right? In Jena and probably Dresden, if I'm not wrong, and 2 are on the way in the UK. So this is the, like, how do I validate it? There's no independent showing this similar. And there's another EMBO paper which you should look at, which has done STED and some EM imaging, which is much better than this one. So let me put a last bit on this, right? So if you have been following STED papers from 2000, microtubules have been the go-to structure, right? And I would challenge the STED and the MINFLUX people who are in the audience to really show me a microtubule which they have resolved something like this. So what you see is one of the latest paper where Stefan is co-author, and this is from Jonas Rees. I don't think I need to repeat, but this is why I really love Jonas. Like, he has one of the best images. So you can see the tubular structure, the ring, hollow ring. So to my knowledge, let's not talk about MINFLUX. Even instead, you have never seen, you can see schematics like this, like 20 nanometer cubes, but it's all full with half maxima and then, decoding back the structure given that we know it. So why didn't authors publish microtubules of MINFLUX, right? Because that's the first structure they do. Anything, I'm pretty sure Stefan has done it. The thing is, microtubules are very dense and they have a very well-defined tubular structure. And if you undersample this based on filtering of localization and points, it will just look a highly discontinuous blobby structure. And then the artifacts of the methods are just visible. So that's why there have been no microtubules. Else, like starting from 98, 2000, whichever paper you look at, microtubule was the first structure showing resolution based on full width half maximum. So this is also kind of lacking, and I still need to see a STED paper before I have desires to see a MINFLUX to really resolve tubules like Jonas has done. And just coming back, a single molecule setup, a very good one. You have prime 95B camera for 1525. You have a good objective lens for $10,000. So you can almost build a very good single molecule setup for 100,000, pounds or dollars, whichever way you look at. Both are equal these days, right? So that was my big point. And last couple of points that I want to make is what happens when you go 3D, right? So in 3D, at least like in 2D, we were seeing, raw filter data, but raw data. As we move to 3D, it's all rendering, right? So we have lost touch with the reality, and now we are in the rendered world. So this is another very important aspect to keep in mind, the 3D. Finally, I say final bit is that Stefan emphasizes that, we have hit a range of 1 to 3 nanometer, right? A range of this resolution. I just want to talk about snap tag, which they use. So you have a protein of interest, then you have the snap tag, and then you have the label. This is snap tag itself is minimum like 3 nanometers. So no way you can claim a resolution less than 3 nanometers. So if you see this 1 to 3 nanometer range, he might be talking about the label, read properly, not the protein of interest. So yeah, maybe label we can identify with 1 nanometer precision. So keep an eye on the word resolution and precision, because in the title, in the abstract, it might say in the range of 1 to 3 nanometer resolution, but in the paper as you transcend, it talks about label, right? So these are very small things that authors do these days, right? So this is more or less end of my talk. This paper is online, the laser-free super resolution, which I talked, and feel free to email me or whatever, and I'm happy to troubleshoot and how you can change a standard setup in your lab to do single molecule stuff. This paper, the MINFLUX, the critique on MINFLUX will be out very soon. I will also do a tutorial, basically inviting all the STED MINFLUX people to contradict what I just said, or clarify my doubts. I might sound suspicious, but it's really doubts. Like if you can clarify me, show me wrong data, I'm happy to take back whatever I have said right now. So this will be on bioRxiv, hopefully, and also online very soon. I would thank Christoph Kramer, Dave Klennerman, David Rovich, who's not here. I forgot to mention him, Johannes, Lothar, Mike, some of my colleagues who helped me a lot over the years and also with this presentation. And thanks for your time and listening.
Well, thank you, Kirti. So we'll go to questions. Stephanie, I guess that you will prepare a Mentimeter. I don't know if now, yes, or would you like to do it now? Oh, okay, perfect.
Yeah, we could. Thank you very much. That was hopefully provocative and exciting, and both at the same time. So
yeah, I hope it was helpful because helpful. Same guys sitting and making decisions. Some people find hard to buy an objective lens, a simple camera, and then you have the same money allocated to tens of MINFLUX. Like already, like 4 of them are on the way. So I think this is quite important, and that's why I have put so much effort in this, because, as I said, research money is not.
Yeah.
I need to kind of have my screen shared for a second, for a few minutes.
Okay. While Stephanie is checking this, maybe. Well, let me do just a comment. I mean, I do agree with the things you said. I also, though, I mean, I do remember when Stefan Hell presented the very first concepts at the focus microscopy. At least is when I saw the first time, and I really was puzzled and I couldn't understand certain things. Also because I do quite a bit of work on photostatistics, and therefore didn't make too much sense what I was seeing. But I must say that over the years, he actually convinced me, and now he sold his techniques to me at least. So I would say that the cost aspect is something that you are rightly pointing out, but this doesn't mean that technology development cannot progress only through cost-effective, accessible technologies.
Alexandra, I completely agree. Like, I think MINFLUX development is super important, and rich labs like yours, Stefan Hell, should invest millions of dollars. But I mean, the core facilities, like I had a STED microscope at IMD. It's 10 years now, and I think first publication just came out. I had a STED microscope at Carnegie. Hardly any publications come out, so it's $1 million of investment. Another thing is, the method is not open source, right? So other labs actually can't work. It's all via a barrier that you will get the access to it. So behind the scenes, like if it's like Lightsheet community, putting out all the designs and things, and it's costly, of course we should develop it. But my point is, if it's being commercialized so heavily, then then we have to, because not the codes, raw data. I think the microscope design is there in the supplementary material, but it's still
yeah. Okay, we can go ahead with the discussion after the Mentimeter, though. Okay. If in the meanwhile the people has other questions, please flag them in the chat.
Yes, so we're going ahead with the Mentimeter, and I hope you. I mean, for $2 million, I would buy. Question is obviously the entry question to the Mentimeter, and some 15, 16, 17, 18, 17 people wanted to will buy a one MINFLUX system. I hope you saw that you can also buy 571,428 fold scopes, and then everything in between. So there are kind of people for every one of the options, which I think is pretty good. But I think we do get a tendency that quite a few people would actually quite like to buy a MINFLUX system.
I'm really happy there are so many STED and MINFLUX people, and I hope you ask some really good and tough questions to me, because even if I'm not able to answer, I will think about this and come back. It's really clearing doubts, nothing else.
Yeah. Alison North is just pointing out that she says, I wish we had been given the option to pick one STED, one SIM, one LSM, one SMLM, because that would give us the correlative approach, which I'm sorry we missed this. So maybe that would be a good option too, obviously.
Yeah.
So let's see. So if everybody has signed up who wishes to participate, I'm going on with
like
the.
I think maximum we had for a time, right? Like people participating. We usually have around 15.
So here, these are the people who are there, and I'm just going to go through the answers. So make sure you answer quickly as well. So these are questions and answers by Kirti. What's the most important aspect of single molecule localization microscopy?
Definitely, you have changed the answers, by the way. Some of them.
Well, I kind of. There you go. Yeah, you were not quite careful enough here, because all of the above were actually correct. 1, 2, 3, 4.
You see, like importance of data filtering, it can give you super highly localized points. So filtering is also important these days, data processing. This first one was a bit tricky. Yeah. Actually, I agree.
Question 2.
It's important, yeah.
Keep doing this.
Definitely, if you get everything right in the first go, my heart will
Yeah,
break.
it would be a miracle, hey?
Oh, no.
Sorry, guys. So here now, question 2. Localization, precision, and resolution are the same. And you have 3 options here. True, false, or ask Rainer Heinzmann.
This is again a smart work by Stephanie. I only had
I worked this a little bit. 5 at least, kind of are risking not winning the full scope by asking Rainer Heinzmann. So,
Sorry, Rainer.
so question number 3. Lasers are essential for single molecule super resolution imaging. True and false. I think if you get this wrong, I think you haven't listened to Kirti 1 minute of his talk. So I think you should be able to get this, true or false. So just a 50-50 chance.
Because apart from my talk, there was nothing to look at, right? I have immersed myself.
Yes, so that's fine. Now question number, there will be 5 questions. So question number 4. This question is, MINFLUX does complex filtering based on, and again, relative photon numbers, background events, both A and B.
So this time you have increased the time for each question, which is nice.
So 7 more seconds to reveal the answer. And again, I think this would have been quite easy for the audience. So 19 have the right answers. So excellent. And then the last question, make sure you prepare this and you get it right and you do it quickly. So which paper do you think had the best 2-color NPCs images? Sauer Lab, Rees Lab, Hell Lab, or all of them are excellent? You can be diplomatic or you make a choice and see.
By Stephanie,
obviously, this is probably. I think there may be quite a few Rees Lab members maybe here in the audience. And careful answer is obviously all of them are excellent. I think even Kirti wanted to be diplomatic here. And so there you go. So these were all the people who played, particularly like both team at both phase, but winning is Mars, Mr. or Mrs. Mars. So whoever you are the winner of this amazing Mentimeter questionnaire. If you send me your email and your address, I think there was one previous winner I haven't actually had sent a full scope. So I think I need to kind of come back to you and get your address. So if you're in the audience, can you also just re-email me? But otherwise I will find you. And yeah, so thank you very much for your participation. How exciting on such a gray and foggy day.
Outside planet Earth, only people from Mars won. So,
so there are quite a few answers, questions.
Yeah, so thank you for the Mentimeter as usual. Is always very nice. I just there are a few questions. So first, I give priority to the others because we can speak whenever we want. So Tanoi, I don't know if you want to unmute and do it by yourself. I give you a few seconds to give a sign. Otherwise, I go ahead. Okay. So does 3D MINFLUX improve the filtered out portions of nuclear pore complexes without losing the resolution much?
Yeah, my guess is, Tanay, my guess is as good as yours. So it's there's no raw data available, but as we see, it's very few points there, and it looks highly undersampled. So yeah, of course, if you choose the best localizations, you will get better resolution also, right? So yeah, I hope like there is enough pressure to really put out the raw data so that the doubts in everyone's mind goes away. Yeah.
So Sherry Huang.
I think Sherry is always joining us, but she's I think
I was checking. Okay, couldn't see
just
her on my screen. So anyway, so talk about label-free.
Oh, there she is.
Ah, okay.
Yeah, sorry. Yeah, because I didn't read. You know, I don't know. It's me.
Yes,
sorry.
yes.
Okay. So I just wonder that because you talk about the label-free image, super resolution image. I always wonder if we have any chance to have some breakthrough if we give up to use intensity as the image contrast, like other label-free imaging techniques. So we can just like another way to break the reflection limitation. And because I don't know, because MINFLUX is kind of a very complicated techniques, it require really I mean high threshold of like engineer and everything. So I just want to have your comment on this topic. So thanks.
So, 2 comments on this. First one, like one part of the talk I showed laser free, where you use the lamp. And I guess by label free, you mean the Hooke's DAPI stuff, where Hooke's DAPI directly bind to DNA and you don't need labels. And there is a 1000-fold enhancement in fluorescence. So I think this is really one of the best techniques to image DNA, probably because there is no linker, right? So whenever the binding happens, there's a 1000-fold enhancement in fluorescence. And this can really also push the resolution. And possibly if you combine this with MINFLUX, you might really actually hit this 1 to 3 nanometer range that everyone is, chasing. I think it's still open race, like 1 nanometer limit. So yeah, with Hooke's DAPI, where you have without the DNA paint. Remember in DNA paint, they actually use Hooke's and DAPI, then you have the protein of interest, right? So again, you have some linker length in DNA paint. But with this, it's published under the binding activated localization microscopy. Jonas Rees lab did the first time, and then we also did with Hooke's DAPI. And there's now Sir Hooke's also. Recently, a paper came out where they again do the same idea, but it has slightly longer linker length. So definitely, like I think I actually wrote a grant, a postdoc proposal on this, which got rejected. But I think with high NA objective and with this binding and binding kinetics, you might actually hit, and it will be also good for these patterned light correlative imaging, whether it's SIM or donut, and you do single molecule, it will be also good because you can very nicely manipulate the binding times.
Okay, so now from Luciano, he was asking if what is your opinion about MINFLUX performance in tracking of fluorescent molecules in living cells as compared to camera-based SMLM approaches?
So I think this is the very honestly speaking, this is the best. The particle tracking is the most let's say exciting application of MINFLUX, I believe, at least in the bacterial samples that they showed in the initial science paper. But my only point would be, do you want to invest so much money, or you can do some tracking in turf modes, for example, right? So yeah, I think that's the best application. And if the prices come down, let's say, in 100, 200,000 range, I would say like, yeah, tracking is the best application of MINFLUX hands down, just because the way it is, right? Yeah.
So then there is Nick Berry had a question. I wonder if you want to, Nick.
Here we are.
Yeah.
I guess I'm just wanted to get at the sort of intrinsic merit of the approach rather than, because obviously your points about resolution and label density and clarity in the literature is all very well made. So is there any intrinsic merit to the method, which is basically for a single sparse fluorescent molecule in the on state? Does MINFLUX potentially offer higher or the same localization precision as capture on the camera? So are you somehow beating the traditional root N limit on localization precision with this method? Yeah,
so I think Nick, again, like, it will be again like a thought experiment how I will answer you. But the base how good your maximum likelihood estimation algorithm is, how well you can combining it with photons. Another thing I forgot to mention. So Stefan, in all his talks, he emphasizes on you need 0 photons, right? For MINFLUX, and on the contrary, most of the MINFLUX papers, they have used one of the brightest single molecule dyes, which is Alexa 647. So in that regard, I'm actually not sure, like, but maybe combined with, so they use like these APDs for detection. So yeah, I don't know, possibly, yeah.
Well, also, I mean, we have I go through one technical question and then go. I will probably pick a bit more, let's say, controversial aspects. So there was Derek that who was asking how he can do turf with an arc lamp.
Yeah, so actually I don't do turf. So it's a full epi illumination, and you can't do turf. So why you do turf, right? You do for optical sectioning, right? You have this 200 nanometers where you have the sample. So these synaptomal complex proteins that I showed you, these are super thin, same like nuclear pores or fibroblast cells. These are super thin. So the sectioning you get is through the sample itself. So you're not imaging thick cells. And the synaptomal complex, they are like, when you do the chromosome spreads, they spread on the slide and there is hardly any, background. So that's how you also minimize with some samples. So it's a nice, good sample. Same as with the nuclear pores, right? And again, coming back to MINFLUX still has to prove on thick samples, right? I don't know if Stefan has really gone through thick cells. So that's another aspect, right? Either you do it through optics or you can use smart samples, as we can say.
Okay, so so just if I can do a couple of questions as well. So you mentioned about the comparison between different techniques, but I think in the original paper from Balzarotti and colleagues, and this is a field I usually don't read literature about. I just know from conferences. Yeah? So you will forgive me if I don't recall properly. But I think when they describe the theory and the hardware implementation in that paper, they also do some side-by-side comparison with other super resolution techniques.
Alexander, can you remind which comparison did they do?
I think they compare with STORM.
Not to my knowledge.
Not with didn't they do origamis, DNA origamis?
So Alexander, there are two papers. The science paper in 2016 and 17, they show the performance on DNA origami, which is a synthetic structure with no background and with underlying structure.
Yeah.
And then the particle tracking they show is on bacterial samples. That's it. And then there's a 90-page supplementary with a lot of maths. In the nuclear pore paper, the nature methods where they do the 3D, it's only nuclear pores. They don't even try to do single molecule, because Jonas was also co-author on that, right? So they could have easily image the same nuclear pores with that, but they haven't done. You can show me, prove me wrong. Maybe I missed a supplementary, and I would accept. Like I would tweet on Twitter if anything I said is wrong, and I'm happy to accept. But
no, I understand. I actually understand what you said. So that's it. Mine was a real question.
I've been literally sleeping with the papers. So.
No, because from I see this from the point of view, and actually something that Stefan likes to say when he speaks about his his journey from not being accepted as these techniques could work to being accepted and receive the Nobel Prize. So he always mentioned that he wants people understand to make a distinction between technological implementation and theoretical background, meaning that sometimes it's the technology doesn't allow you to do something, but in theory you can do it. And I think what is important about MINFLUX is that at the moment it shows another way to potentially reach, molecular resolution.
Alexander, I completely agree.
Oh, sorry. Just to finish, just to say, I would like to see if to understand if there is something that in the theory that is considered, unachievable, put in this way.
No, I said it before and I say it again. I think it's beautiful physics, and places like Janelia Farm, which have like $500,000 lying around and they don't know how to use it, or Stefan Hell Lab. So big lab should push, But my whole point was, DFG or ERC or an IEH, instruments being bought, which still I believe haven't proven themselves, and from public funds, right? So that's my thing. Like I have No exceptions. I think it's a good method, very exciting. I like to think about it, and if I have $100 million, I would put 10 people on MINFLUX, because I think it might be the next big thing, right? If imagine we see atomic structures with MINFLUX at some point, right? So you never know, because you need 0 photon. So that's what I said initially. I have no criticism on technical side, partially because I have never worked with the system. I have never seen it. My only thing is, what do we get at the end of the day with the images? If I want to do biology, and if core facility wants to buy it, MRC, LMB, cancer research. I was part of one of the grants where we proposed buying MINFLUX. So like all these grants which don't come like from big labs.
No,
that's my only point.
Yes, yes.
Critique. Yeah.
There is a question. Okay, there is a question from Nathan Curry. He say the Minstead paper implies that MINFLUX struggles if the intensity at the minimum of the donut is non-zero. Similarly, if there are aberration background. I think they also do some maths in the Nature paper on how these affect the precision.
Yeah, absolutely agree, and I think it's an important comment. It's a science paper, I think, but and Nature methods. Yeah, definitely. So yeah, I just agree with what Nathan says. So and especially like you have to still check the performance of MINFLUX in thick samples where there's more background, and with so if you see, they have used Alexa 647, right? And also for the live, they have used one of the brightest dyes. So I think as you go down to more, let's say, like where you have GFP integrated with your system, so something like that. So Jonas Rees lab, for example, right? In this Nature Methods paper, they projected around 7-8 cell lines in total for this nuclear pores. And I would like to see how MINFLUX perform with different labels, right? Different labeling strategies that Jonas have, but they just show one of them, which is a snap tag with Alexa 647, which is the best dye. Yeah.
I don't know if you want to comment on something that an old friend of mine, Mika, also originally we met in Göttingen a while ago. He was hinting to these discussions about that there was a technique around that that based on the same principle. I don't know if you can comment on that.
You mean Steve Baer work, and there is also Gabriel Sirat, which is in France. I'm just missing the name of the company. Yeah, so there are a couple of Steve Baer talks. It's, I think, 2010-2014 patent. I have tweeted it before. The ideas of MINFLUX, and then Gabriel Sirat. He's most famous for conical refraction, and yeah, bioaxial. Thanks, Mika. So I think there is a paper by him, again, like 6-7 years ago. I think I have tweeted that also some year back. So I think these two methods are very close to MINFLUX. I also know a theoretical paper, a very old one by Reiner, where he discusses this maximum likelihood ideas on this. So there have been more efforts, but yeah, they haven't been in public domain. Yeah.
Well, I also remember that there was a work, which is the first thing that came to mind when I saw the first time MINFLUX. Enrico Gratton was doing this tornado scanning to track, but that was for tracking, particle tracking.
Exactly. So from that lab, actually, I asked Michelle Digman, because she was in his lab around 2006, 2008, and actually that was one of the first ideas, using a MINFLUX idea for tracking. So yeah, if you can find that paper, I would be very grateful, because I was searching for that paper like 6 months back, but I could not find it. I think it was just an abstract or something.
Yeah, again, I saw it in talks, I believe.
That's spot on. I think that might be actually the first application of MINFLUX. Yeah.
Yeah, but of course, that was not. It wasn't with the donut, and it wasn't for achieve the same resolution. But the concept is it was
same, yeah,
nicely tracking. Yes. Okay, is there any other?
Yeah, so Peter, mentions this conical imaging exactly, yeah. So.
Okay, if there is no other questions, I guess that I can say, Stephanie, if you want to take over. or Alex?
Stephanie's gone, so I think thank you everyone for coming through. Thank you, Kirti, for excellent.
Alex, we don't hear you. The microphone need to go
It's up
down.
here.
Sorry. So no, I was just saying thank you very much, Kirti. That was a great talk, and thank you very much everyone for coming along. So I think next week we have one final talk before Christmas, and I've just forgotten who it is, but it'll be advertised in the near future. So yeah, thank you very much, and I'll give an embarrassing clap. Everyone can clap now, or do some other virtual reaction.
Yeah.
Thank